Yeah. So what we were talking was about nucleic acids. Nucleic acids are polymers of nucleotide and nucleotides are nucleoside plus H3PO4 and nucleosides are a sugar plus a base. Sugars are ribose and deoxyribose. And bases are pyrimidine bases and pyridine bases. Pyridine bases are adenine, guanine, and pyrimidine bases, thymine, cytosine, and uracil. Okay. So this is where we have stopped last time. I think we have written the structures. Now let us make a nucleoside. We'll make how to make a nucleoside. What is a nucleoside? We require a base. Okay. We require a base and a sugar. We'll take the ribose sugar. This is a ribose sugar with CH2OH here. And then OH down over here. So we have kept the OH. Then here the base is getting fixed. So this is so you have the two nitrogens, one is here, one is here, 1, 2, and 3 double bonds, it's aromatic, and NH2. And there is a double bond here. And it is attached to nitrogen. Now see here, this molecule is adenine. This base is adenine, and this is ribose sugar. So we call this as adenosine. We call this as adenosine. So adenine plus ribose is known as adenosine. Now another one. Again the same CH2OH. OH. And there is nothing here. It's hydrogen. On the next carbon you have hydrogen only. And then we have attached here a compound of this structure. And here and a double bond here. SCH3 here. CH3 and two keto groups here. There is only nitrogen. And this nitrogen is having a hydrogen over there. NH. Now this is deoxy ribose sugar. So we can write deoxythymidine. Deoxythymidine. Similarly, you can draw any of these nucleoside structure. So these are just the structures of two nucleosides. Okay. Now, one more thing you can remember, you can note that ribosthymine ribosthymine that is this is ribose adenosine, right? The first one is ribose adenosine. Ribose connected with thymine is not seen, not observed. Okay, that combination is not found in the nucleic acid. Similarly, deoxyribose uracil, deoxyribose uracil combination again is not observed. Okay, this combinations are not. This is another way of telling you. This is a second way of telling you that DNA contains the four bases A, T, C, G, and RNA contains A, U, C, G. That's all. These two statements are same. You can observe it. See here. DNA contains deoxyribose, right? DNA does not contain ribose, it contains deoxyribose. And see here, you don't have uracil here. So this is a second way of telling you that DNA contains only these bases, RNA contains only these bases. And here you can see here, RNA does not contain thymine, it contains AUCG. So this is just a different way of telling you which bases are present in which nucleic acid. Okay, let me just switch on the light. Yeah. Okay. Now, so this is now let us talk about nucleotide. So this is nucleoside structure. You have to remember it is at this point that the base is getting connected on carbon number 1. So base is connected here and here. And base is attached to the nitrogen atom. Base is attached to the nitrogen atom which contain the proton. Okay. So these are the things which you have to remember. Now let us draw the structure of nucleotide. A nucleotide. So we will take this nucleoside down. So here is a nucleoside. Once we have this nucleoside, and what is the base? We'll take this base. Let us complete that structure. 1, 2, 3, and NH2. Lot of nitrogens. See that? Okay. And then we have the OH here and CH2OH here. So this is yeah, you have to. So this is adenosine. Now let us combine this with a phosphoric acid molecule. Combining this with a phosphoric acid molecule. Now let us look at the chemistry of it. An acid will react with alcohol. To give an ester. This is what is going to happen here. So here is alcoholic OH. Primary alcoholic group is more reactive with an acid. And what we are doing is the OH, and let me draw the phosphoric acid here. OH. Yeah, then we have H3PO4. P double bond O, single bond OH. 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 Now see here, this is one molecule of phosphoric acid, this is one molecule of your nucleoside, adenosine. Now what we do is we take one molecule of water from here. We remove this water. Once you remove the water, what you will get is the ester of it. So we have connected this here. Remember, it's not a long bond, you have to draw it closer. Okay. So this is the structure of a nucleotide, and this is called adenosine monophosphate. Adenosine monophosphate. Adenosine monophosphate. Okay, so you can draw the structure of any nucleotide like this. Okay, you can construct the deoxy also. You can use this and construct a deoxy here by attaching a PO4 here. Here you can attach a PO4. See here, P double bond O. It's so easy. OH and OH. I have removed the water from there. Okay. So this is the structure of nucleotide. Now we have to draw the structure of nucleic acid. Now, how do you draw the structure of nucleic acid? Now, what is nucleic acid? It's a polymer. So you have to polymerize this. Only difference is, the difference is in usual polymers, which we make in the laboratory, it's a repeat unit. Repetition of same unit or multiple one or two units. But here you have a lot of possibilities. Adenosine can combine with thymidine or uracil deoxy, that is guanosine. Any combinations are possible in the same nucleic acid. So that is the difference. And it is very specific sequence of the DNA. So let us construct the DNA molecule. So what is DNA? In brief terms, you can say we have a phosphate. I'll write phosphate for this one. The phosphate is combined with the sugar molecule. The phosphate is combined with the sugar. What is the sugar? Ribose or deoxyribose. Then on the sugar, a base is attached. And then on the sugar itself, there is another attachment to the next phosphate group. Attachment to the next phosphate group. And then it gets repeated. You get the next sugar. Next sugar. From the sugar, on the sugar you have the second base. The second base. And again, this sugar molecule is attached to the next molecule of phosphate. So it gets continued like this. Now we will construct the full structure of a molecule of DNA or RNA. Now you have to polymerize this. Now the combination is over here. This side is getting attached to the next sugar molecule. So let me draw one of them here. And then I have got a CH2OH here. And O, P, double bond O, O, H, P, O, H here. Now this is further getting attached to the next sugar molecule. Next sugar molecule, which is again, you have the OH here, OH here. And you have the base here. Base. And here also, we have an attachment here to the next phosphate group. Now combine this with the next molecule of sugar through the CH2OH bond. Once, so went off my hand. O, CH2, and this goes to the next sugar molecule. Okay. Again, you have the third base here, the OH here. The next molecule is getting attached over here. So you can see this is 1, 2, 3. 1, 2, 3, 4. 5. So this is 5. This is a combination, 3-5 combination. See here. This is the third hydroxyl group on one unit get bonded to the sixth carbon on the next unit. Okay, see, this is how you can draw the structure of a nucleic acid molecule. Okay, it is beautiful to look at it. It is step like that. So this is nucleic acid. Suppose you are asked to draw the structure of DNA molecule. This is RNA, of course. If it is DNA, what you have to do is you change these oxygen. This oxygen, this oxygen, this oxygen, you change it into hydrogen. So simply remove that oxygen. Number one. Number two, your base should not contain uracil. Base should not contain uracil. Okay. And one more thing. DNA is a double-stranded helix, which we are going to discuss. Okay, that we are going to discuss. That's all right. So this is how you form the backbone of the structures of RNA and DNA. Okay, so make sure that you learn this because these are very useful for you people. Now, the characteristics of DNA and RNA we have already mentioned quite a few. DNA is double-stranded, RNA is single-stranded. Okay, and in RNA molecule there will be folds in the chain, different type, different fold, F-O-L-D, turns and folds in the chain of DNA and RNA, which are very characteristic of the nature of the material. Now we have different types of RNA. The names of different RNAs we call as messenger RNA. Messenger RNA. Okay, it is also called template RNA. Template RNA. Now, what is a messenger RNA? Now, the messenger RNA is a single strand molecule which is having a sequence complementary to its parental DNA on which it is synthesized. Now, here is a DNA having a double helix structure. This DNA is responsible for the construction of an RNA. This DNA synthesizes an RNA. Okay. And the sequence of the bases in the RNA will be complementary to this DNA. Now, what is meant by this complementary? The simple thing, the combinations of bases are adenine will bond with thymine. And this is hydrogen bonding in nature. And cytosine will bond with guanine. Okay. Wait a minute. Hello. മനസ്സിലാവും ഞാൻ വിളിക്കാം ക്ലാസ്സിലാണ്. Okay, so adenine will combine with thymine and cytosine will combine with guanine. So this is what you have to remember. So if you write a sequence, the sequence of bases of a DNA, here, over here, sequence of base, say for example, A, an adenine, then T, then you have again A, now G, and then which one, which is not, which is missing, a C and T, again G. Now you can draw the structure of the RNA here, which is complementary to this. Now see here, A, which is complementary to A, T. So I will write T here, which is complementary to T, A. See here. Again A, T. Now G, what is complementary to G? C. And for C, it is G. And for T, oh, I made one mistake. Something which I have forgotten is, see here, that is very important. In RNA, there is no thymine, right? In RNA, there is no thymine. So complementary to thymine, sorry, complementary to thymine, it is AU. In RNA, you have, there is no T in RNA. So you have to write in the place of T, you have to write U. Yes. See here. Wherever you have T, should write U, uracil. Okay. So for thymine, it is A, adenine. For guanine, it is C. So this is the structure of the complementary RNA to this DNA, right? So this is how you write the structure of RNA. Now, why did I write this? RNA is synthesized from DNA. DNA is the basic unit in the cell which transfers the gene. And during the protein synthesis, this is the first step. During the protein synthesis, this is the first step. What is the first step? The DNA carries the code of the protein. This DNA carries the code of the protein. And it is transferring that code through the messenger RNA. So this DNA is making a messenger RNA here. It is making a messenger RNA. Okay. And this messenger RNA is moving out of the nucleus and transferring this data to another type of uh, RNA, which is called transfer RNA. So first one is messenger RNA, which I have described how it is being made. And the second one is transfer RNA. Okay. Now, the second one is transfer RNA. No. The transfer RNA is a single-stranded RNA. Okay. And it is a very small polymeric form of RNA and having a molecular mass of about 25,000. Okay. That's, and it is soluble in cytoplasm. The transfer RNA is also called soluble RNA, and it transfers the code of specific amino acid from the pool of amino acid in cytoplasm to the correct site of ribosome for protein synthesis. So, in the cytoplasm, there are so many amino acids like glycine, leucine, valine, alanine, then what do you have? Histidine. All these amino acid is there in the cytoplasm. So what transfer RNA do is, transfer RNA will go and pick, see here, it picks the suitable amino acid and carry them and give it to messenger RNA or